of the last. And now in this presentation, we are going to talk about use of the human umbilical cord, mesenchymal stem cells, and exosomes in the treatment of liver fibrosis and cirrhosis in Wistar rats. So we did this animal trial in India. And again, I'm Dr. Boddu from San Diego, California. My disclosures are I'm part of Tulsi Therapeutics Limited, and this is my clinic in San Diego, California. So we talked about this adult bone marrow stem cells sourcing and why we chose mesenchymal stem cells in the last lecture, and we know the pathway, how they work. So the mesenchymal stem cells, we talked about them getting engulfed and becoming the regenerative stem cell from immune cells. Now, we also use exosomes. Now, exosomes are the small little vesicles that mesenchymal stem cells produces. This shows the pathway of making exosomes. They kind of, in the beginning, they start as endosomes by the mesenchymal stem cells. Now, remember this, exosomes are made by pretty much every cell. But the question is, do they have a regenerative factors and vesicles and or not? And the difference is mesenchymal stem cells, exosomes have those. And they can take inside they are called multivesicular bodies. And they can either go the lysosome pathway, or that's destructive pathway, or the exosome pathway. So now again, exosomes by the International Society of Extracellular Vesicle definition, they have to be released by the cells, and they do not have nucleus. That means they cannot replicate, and they need to have the lipid bilayer. So all the vesicles released by the cells, again, they are depending on their size. 1,000 to 5,000 nanometers is apoptic bodies. Between 100 to 1,500 nanometers are called microvesicles. And then the exosomes, which we are interested in, they're about 30 nanometers to 150 nanometers. So what do they have? They're secreted by the mesenchymal stem cells. The exosomes of interest for us, we are looking into the exosomes we used were the umbilical cord mesenchymal stem cells of humans. And when we extracted the exosomes from them, what do they have? They have nucleic acid, as we talked about. Like I said, every cell makes it, but the one we prepared are from the UCMSCs. They are part of the cell-to-cell communication, and they are cell-specific, and that's why the factors we have are specific to mesenchymal stem cells. This is almost like a liquid biopsy of what the cell, where they came from. So now they have multiple things. They're cytoskeleton proteins, they have growth factor cytokines, nucleic acid, mRNAs, microRNAs, single-strand DNAs. What's important is tetraspanins. Tetraspanins, one, and they are the one which helps them guide. And they have some antigen molecule presentation too. And then the other thing is the annexin and flotilins, and these are important for, we'll talk more about down the line about that. So like I said, the functional components are the nucleic acid, the growth factors, the receptors, and the organelle. So like I mentioned, it's a phosphobilipid, the enclosed by lipid layer. And so this phosphobilipid protects the contents of the exosomes for about 3 hours in the medium. And what also it helps is to recognize the target cells. We talk about the homing. That's what is happening. We are recognizing the target cell. And then they're going to the target cell and binding it. They're recognizing the sick cell, the apoptic cell, and they're helping them out. So like I mentioned, they also are tetraspanins, and they are the CD963. They're required for target recognition also, and most important, attachment into the target cell, the sick cell. And then, as I mentioned, the annexins and flotilins, are important for entering the target cell. And then once they enter, they release the whole cargo, all the contents. They're going to release that into the cell. So now the uptake, it's active uptake. It's energy dependent, and then it's temperature sensitive. So once they find the target cells, exosomes, they go and dock. And after docking, the exosomes gets attached to the cell membrane, and then the whole exosome gets inside, becomes the intracellular content, and releases its payload into the cytosol of the target cell. So this happens to the, what do you call, membrane fusion. So this is a transmitting electronic microscope transmission, and this is how you see the exosomes. This is pretty cool. Like, we could really see these little things rather than just imagining them. And so this is this is one of my favorite slides. So this is one of the MSCs, mesenchymal stem cells, and you can see the formation of. You can actually see the pathway. You can see a pathway of their multivesicular bodies, and then how they kind of, from there, they go either the pathway of lysosome formation or exosomes. And here you can see the exosomes getting out of the cell. I mean, if you have to just illustrate it, it's going to be like this. So that's a mesenchymal stem cell. And it forms early exosomes, later exosome is formed, and then from there it forms the multivesicle bodies. From multivesicle bodies goes the pathway of degradation, lysosomes, or regeneration, which is exosomes. So the pathway, it has a similar effect. It works through the immune system. It converts the pro-catabolic cells, again, the immune cells, to a regenerative cells, and then has a whole micro-anabolic microenvironment. So the tissue injury, we all know about tissue injury. The inflammatory cytokinins, interleukins, necrotic factors. So the normal cell, when it's in danger or has sustained considerable insult, and now it's going to die, either takes one of the two pathways. It either goes towards the apoptosis pathway or necrosis pathway. In the apoptosis pathway, it's a little different. The membrane becomes blebbing. The little membrane, because all the blebs are formed in the cell, and that's how the cell is going to die. And then after that, the cell gets broken apart. It ends up forming apoptic bodies. And what happens is the phagocytes just chew them up and say they don't want to create any inflammation. So in this pathway, you don't have inflammation. Now the necrotic pathway, the cells, they swell up. It takes up more water, fluid, protein, and swell up and stuff. And basically, cell membrane ruptures. There's a cell membrane rupture, the nucleolysis, all this inflammatory substances are released in, and that's what causes the inflammation. So the exosomes, how do they work? They work two different pathways. One is to modify the immune cells. We talked about this already. And the second is they enter the disease cell. So similarly, like I said, the pathway of injury to repair is influenced by regenerative cells. We talked about this, and exosomes. So you have both regenerative immune cells and mesenchymal stem cells. They produce extracellular vesicles, and exosomes are part of them. In addition to the vesicles, it also produces secretomes. It also has other regenerative factors. They have paracrine effect. They have cell-to-cell signal. So these are all the pathways that a regeneration process occurs. So if you look at the contents, what are the contents of exosome? The most important contents are like I said, single-strand DNAs, double-strand DNAs. They are microRNA, mRNA, and these are the flotilins they use for surface markers and attachment. And they have lipids. If you look at they're proteins, microRNA, mRNA. So when exosome goes and joins and fuses with the target cell membrane, the entire payload of this thing, including the good factors, growth factors, gets released into the cell. And that's how it makes it into a regenerative cell, helps the cell to repair and regenerate. So we talked about this docking and then joining the cell fusion of the membranes. So now we are to see this was a paper published in 2012, basically saying that human mast cell exosomes and the transfer to blood CD34 progenitor cells. That's basically stem cells. Approximately 1,800 mRNAs are in exosome, which basically represent 15% of donor cells. I mean, if you look at that, donor cell mRNA, look at that, that's like a big percent of a capsule of regenerative materials sent out of the uh, progenitor or mesenchymal stem cells. So this is the analysis of what do they do, microRNA. So they cause angiogenesis. They actually have cardiac hypertrophy remodeling. They have inflammation, anti-inflammatory, neuroprotective regeneration, tissue repair. And obviously we talked about M1, M2 exosomes,
Mr. Boddu, you have muted yourself. Can you just unmute yourself? You have muted yourself. Can you unmute? No, you're still muted.
how is it now? Now it's good?
Perfect. Yes, perfect.
Perfect. Sorry about that. All right, thank you. So let me get back to where we are. There you go. So yeah, similarly, the B cells, the regulation of B cells. So that's another thing, pathway it goes with. Again, dendritic cells, the antigen presentation capability decreases that, decreases the maturation, and then becoming a catabolic pathway, inflammatory pathway, and conversion of D cells into conventional D cells. So now, what happens to exosomes? When you give exosomes to the patients, now this is a beautiful representation in different organs. So the first hour, obviously, liver gets a lot of it. The red is all lot. Liver, and then spleen, lungs, and kidneys. They get the most of it. As the time goes by, kind of, you get after a few hours, your GI system gets a little bit more, and the bladder. But about after 24 hours, there's still a good number of exosomes in the liver, also spleen and lungs. It's interesting. Check this out. The pancreas starts picking up more exosomes. So this is exosomes again. That this is what we call infusion intelligence, where you infuse or give the medication on healthy pigs. Not much are taken up. A little bit in the kidney, but not much after 5 hours, but it's gone. But on a patient, mouse with acute renal kidney injury. But look at the way the concentration on this after 24 hours. That's an amazing. And this is the same way the exosomes. Uh, mesenchymal derived exosomes on mice with ischemic stroke. Day zero, not much, but look at by the to day 10 or day 14, and that's what I call about the infusion intelligence. So we talked about the T cell regulation. So this is our background. So this is a background for our animal trial we did in India with Tulsi Therapeutics. So what we did was we used the mesenchymal stem cells from umbilical cord for their anti-inflammatory properties and regenerative properties. And in the past, this has shown in animal models to for antifibrotic effect and decrease the liver fibrosis and cirrhosis. But ours is the first study ever done. We examined a combination of human umbilical cord mesenchymal stem cells and combined them with we call it small extracellular vesicles, basically exosomes. And what we saw was the effects of liver fibrosis and liver cirrhosis in the rat models. So we picked the rats. They're about 7 to 8 weeks of age. There are two groups, a control group and treatment group. And what we did was we used 14 animals in each group. And we induced liver failure in this rat, a chronic liver failure model using carbon tetrachloride. Both animals received the same dose, induction dose of CCl4, and then so the oral card CCl4 was given with olive oil, so that the rat ate them up. They were basically two times a week for 6 weeks, and then from week one to sixth week. So there were 14 animals in treatment group, as I mentioned. And then what we did was intervention was in the treatment group among these 14 animals. We started in week 4. We infused them IV dose of umbilical cord mesenchymal stem cells and exosomes. Now, so we infused this every weekly. They got doses. So 3 weeks they got total 3 doses. So they got 1 million mesenchymal stem cells and 5 billion exosomes. And the mesenchymal stem cells they received were three passage 3 human umbilical cord mesenchymal stem cells. They were provided by Tulsi Therapeutics in India. Now, our objective, what do we see? We want to see the survival. We want to see the routine liver test. We want to see liver fibrosis, like what's happening with the liver fibrosis, and see the liver cirrhosis. Is there any, how bad is the cirrhosis? And then, is there any liver regeneration? That's more important. So we see that. So at the end of 7 weeks, after we started giving the CCl4, all the live animals were sacrificed. All right. So what did we see? So in the control group, before the week 6 animals died. So in the treatment group, none of them died. And in the control groups, they were losing weight. They were reducing their feed intake. Their livers got larger, by size. And the necropsy of dead animals showed liver cirrhosis in all the dead animals. These are the animals that died before 6 weeks. So, and then after 7 weeks, we sacrificed 12 animals had the cirrhosis. Okay. Now, what did we see in the treatment group? None of the animals died. They were normal weight, or even actually they gained weight. And then their feed intake was normal, or some of them were eating voraciously. And their livers were normal. And then, well, none of them died, so that didn't apply. So so we looked at the animals, and none of the animals had cirrhosis. So and moving forward, there you go. So then the control group, 8 animals, and 14 animals in treatment group. Like I said, they were sacrificed on week 7. And this is a very busy table, very busy table. Look at the p-values of this table. I mean, apart from the body weight, but the very significant p-values. So we looked into what do you call feed intake. We looked into serum. What was amazing is when you come down to the histology, the fibrosis scan, the red, serous red, trichome. We had cirrhosis, like how much percent. IHC, the liver. I mean, this is like amazing, the p-values, and obviously the mortality percent. I mean, in the control group, 43% of them died, and zero. So now we went to the histology slides. We want to see, we see that the previous table look amazing. That's what numbers, but we want to see what happened. I mean, let's look at the treatment. That's very close to normal. Look at the liver. There's absolutely like cell vibrant. The cells. Look at the H and E staining, amoxicillin, eosin dye staining. I mean, look at the cells and regeneration compared to the one with the control group. I mean, there's huge fibrosis scars, the cell death, with all the white type. So that's the HE staining, and then this big take up, you know. So the group of that, and then we did the this is the liver fibrotic score, the same thing. Look at the vibrant cells and what was amazing with the slides was this is regeneration. The slides show this is something we did not expect. I mean, this is something in the past we didn't see. But look at the fibrosis scars and cirrhosis of this liver with the Mason trichome staining. Similarly, the same thing like Cyrus red. Look at the red intake. That's over the fibrosis. And compared to the treatment group, I mean, it's barely red. I mean, look at the amount of fibrosis in the slide. One high power field. I mean, this just was like we were amazed at the regeneration that this treatment group had. And this is the immunohistochemical. This is the SMA, alpha SMA. So similarly, same thing. Look at the nucleus of the cells. I mean, just so vibrant regeneration compared to the cells in distress, swollen up cells, fibrosis, and then this is again the different collagen staining. Absolutely, very little, absolutely no collagen, no collagen. If you look at this slide, compared with the control disease slide. So in every, I mean, this kind of regeneration was practically undocumented in the previous studies. So we were very encouraged for the animal data for this slide. Now we're down going to cytokeratin 19. Same story. I mean, it repeats. So what did we conclude with the study? We said cultured human umbilical cord MSCs and exosomes significantly improved. We've seen that in survival of animal receiving CCL4 and a chronic liver failure picture, and it dramatically reduced the fibrosis and cirrhosis on these little rats. I mean, that's amazing. We're amazed at the amount of regeneration we have seen. Obviously, we need to further study to validate our observation on the animal models and also humans, the fibrotic liver disease and liver failure. And the human, we need human trials to confirm this use of combination. Uh, Again, thank you very much for your time. I really appreciate giving the presentation for you.